Good morning, everyone. Our today's topic of discussion will be what a cell needs to do before it goes for division, or how the cell prepares itself before it starts dividing. Well, every cell that comes to life, it divides, that we know. But before division, there are a lot of functions, a lot of tasks that it needs to complete before it will be ready for division. Now, in order for it to divide, the list of tasks that it has to do are synthesizing RNA, synthesizing protein, are duplicating the DNA, then preparing itself to split. So there are a lot of tasks that it needs to do before it thinks of dividing. And all this task that it does, it performs it in a very organized and predictable series of events. And this series of events we call as cell cycle. Now, the cell cycle can be divided into two particular phases. One is the dividing phase, where the cell division takes place. Another is the non-dividing phase. It is this non-dividing phase where the cell does all these functions that I mentioned, like synthesizing RNA, protein, duplication of DNA, increasing in size, getting ready for splitting, and all. This stage of non-division, this non-dividing stage, when the cell prepares itself by doing all this work, that is further divided into some stages. As I told you, it's a highly organized process. So the cell's work is cut out, that at this stage, it will be doing this particular work. At the other stage, it will be doing another particular work. So this entire non-division phase is again or further divided into various stages, or rather 3 different stages. Let me show you that. What are the 3 different stages, or how the cell cycle is divided? You can now see it that you can now see on your screen that this is a cell cycle, and this cell cycle you can see is divided into 4 stages. This mitosis is actually the dividing phase, and all the other phases that are mentioned here, these are the non-dividing phase, which can also be called as interphase. This non-dividing phase is also called as interphase. Now, this interphase, as I told you, is divided into 3 more stages. So these 3 more stages are first growth phase, which is also known as G1 phase. Next is synthesis phase, which is also known as S phase. And the third one is second growth phase, also known as G2 phase. Now see, here is mentioned that the first growth phase is used for growth and normal metabolic roles. The synthesis phase is used for DNA replication. And the third is used for growth and preparation for division. Let us go a little deeper into this first growth phase. In this first growth phase, the synthesis of RNA and proteins take place, due to which the volume of the cytoplasm also increases. Now, in case of animal cells and in case of plant cells, there are 2 different organelles which have their own DNA. In case of animal cells, it's mitochondria. In case of plant cell, it's chloroplast. As they have their own DNA, so in this G1 phase, in this first growth phase, in the late growth phase, late G1 phase, just before the synthesis phase, the cells decide that which path they will follow. Now, what is that? Which path they will follow means what? It means that After completing the RNA synthesis and protein synthesis and the volume of cytoplasm increasing that, now they decide whether they will divide or they will stay in a resting stage. If the cell decides that it will stay in the resting stage, then it does not proceed further to the synthesis phase. But if the cell decides that it will go for cell division, then only it enters the next phase, that is the synthesis phase. I hope that's clear. So the cell, let me repeat, which does not go for division, which decides to not go for division, they withdraw from the cell cycle and enter a resting phase. But the other cells which decide upon division, they come to the synthesis phase. Now, let us see what happens in the synthesis phase. As it is written here that DNA replication takes place, actually that is what happens here. Your DNA gets replicated. So let me tell you one thing, that we know that all of our cells have 46 chromosomes. Every cell contains 46 chromosomes in a human body. Now, if the cell divides, then as we know that both the daughter cells will get equal share of genetic element. So that means the two daughter cells that are formed, if the DNA is divided equally, one has to get 23 and other has to get 23. Now, if they divide further, what will happen? Will they get half, like 0.5 of something and 0.5 of something? That's not even possible. So in this, that's why what happens first is, before the cell divides, duplication takes place. Before the cell divides, there is a duplication of DNA that takes place. It means whatever number of chromosomes that are present in the cell, they double up and then they divide. So the 46 number of chromosomes that are present, they double up. Now when they divide, they are equally divided into two different cells, and each cell get 46 chromosomes, keeping our number of chromosomes intact throughout every cell that is divided. Okay, so let's see now what happens in the S phase, or how this DNA gets duplicated. So in the S phase, as I was telling you, yeah, bear with me till I bring it. Yes. So this is what actually happens. What is this? Each DNA molecule duplicates. Let us look at this double helical strand of DNA. Okay. So for replication, first of all, this double helical strand, which is so tightly coiled around, which we had learned in our previous video, that opens at one end. That means it starts decoiling, making the strands free, or making the strands available for the process of replication. So when they are becoming free like this, a new strand complementary to this strand will start forming, and another new strand complementary to this strand will start forming. Okay, now what do we mean by complementary, or how do I know it's complementary? We know it by the kinds of bases that are present. If, let us talk about this as the original strand, obviously it is the original strand. So let this be the original strand. Let originally the first base be A here, the second base be T here, the third base be C. So the new strand that will be forming will follow the rule that I told you last day, that A will only bind with T, G will only bind with C. So as the first one was A, so the new strand that will form will have a T over here that will be joined to the A of the previous or first DNA strand by a double bond. Then if there was a T, then the new strand will have an A. Then if there was the base guanine, the new strand will have cytosine, like that. So the new strand that will be forming will be complementary to the first strand. Here also another new strand is forming, which is complementary to this strand. And henceforth, these strand, the decoiling of the original DNA strand will take place, and the formation of the new strand will take place. Okay, this will take place throughout the whole sequence of the DNA. This will take place throughout the whole sequence of the DNA, at the end of which we will get two separate new DNA strands. Okay, so now this happens inside every cell that goes for division. Now, what I told you as replication is a highly simplified version of DNA replication. It's actually a very complicated process, which obviously you will study, but not now, in higher classes. So that ends our S phase. S phase is up to this much. That ends our S phase. Now the last phase that we were talking about in the interphase, it's G2 phase, or the second growth phase. What happens here? In this phase, it is shorter. It is a shorter phase in comparison to G1 and S phase. Now here, the RNA, the proteins that are necessary for the cell division, that continue to be synthesized in this phase. Okay, and it is this phase where the cell is completely ready for cell division, and after this phase, the cell moves to cell divisions. The cell moves to mitosis. Here I am referring to mitosis. We'll learn about a second type of division later on. The cell moves to mitosis after G2 phase. So this is the entire process of cell cycle. Let me have a quick recap of that. There are two processes, or rather there are two phases of cell cycle. One is the dividing phase, another is the non-dividing phase. The non-dividing phase, we also call it as interphase. We have focused here on the interphase. It has three stages: G1, S, G2, G1 being the first growth phase. In the first growth phase, what we do is, the cell does is, it does RNA synthesis, protein synthesis, it increases in size, all these work. Rather the the volume of the cytoplasm increases. And at this phase, the cell also decides that whether it will, after this RNA and protein synthesis, whether it wants to rest, go to a resting phase, or it wants to go for division. Once it decides that it wants to go for division, it commits itself to division, it goes to S phase, and then there is no turning back. In S phase, DNA replication takes place. So whatever DNA you have in your body, in a particular cell that is going to divide, gets doubled. Okay, so it can be called DNA duplication, DNA replication. Okay, next comes G2 phase, where the RNA and the protein that is needed for cell division, that starts synthesizing, or that continues to be synthesized. And then the cell enters the cell cycle. Now, a question might arise that if this is the process of cell division, then can the cell division continue forever? Like the cell cycle can go on endlessly. like one cell divides into two, two into four, then keeps on dividing and dividing and dividing, and without any stop. Or who, or how to stop this cell division? Who decides to stop? Let me tell you, there are the cell division is not, it does not go on endlessly. The cell cycle does not go on endlessly. It stops at some point. It stops temporarily, and at some places it stops permanently. Till at where it stops temporarily, till it is needed again, it pauses there. Now, there are many cells in our body, different types of cells in our body. The cells such as brain and nerve cells, they are formed in the embryo itself. And they, once they are dead, they are not replaced. Similarly, there are liver cells which divide only 1 or 2 years. But there are red blood cells. See, after every 120 days, a red blood cell dies. So a new cell needs to be produced. So their division goes on at a higher rate than if we compare it with liver cells. So different cells have different rate of division. Some cells do not divide at all. Then who decides this? Actually, at the end of every stage, G1, S, G2, there are checkpoints. There are checkpoints which decide whether the cell has done all the tasks that it needs to finish before entering the next phase, whether there is any fault, whether there is any problem that it faced, whether it did not do the task properly, whether the RNA and protein is not synthesized properly, whether there is a problem in DNA replication. They see through that, and if such a kind of problem is found, anomaly is found, the cell is not allowed further to go into division. So these checkpoints are very important, because if there would have been no checkpoint, the cell would have started dividing non-stop. And or if the checkpoints were faulty, the checkpoints were not doing their work properly, then a cell with maybe not properly replicated DNA would have been able to divide. That would have led to, or these problems lead to, tumors that may or may not be cancerous, but also the checkpoint failures are a huge reason for cancer. So what we learned today is that though there is the cell cycle to help the cell to go for division, the cell cycle does not go on endlessly. There are stops, there are checkpoints, depending on the body's need, all this happens. Okay, so that's it for today. In our next video, or in the next class that we'll talk about cell division, mitosis and meiosis, what happens in this. Okay, so thank you for today. Have a good day.